Are you posting the other ones from the last few weeks?
Yeah, I lost the first one, but the second one is posted. I thought
Okay.
I could have sworn I posted the first one, but I can't find it anywhere. I can't find the file,
Mhm.
um, so I just don't know. Uh,
Okay.
but
I'll
the
just call
second one
you.
was posted.
I'll just give you a call at midnight when I need help.
So as per usual,
I
Yep.
have a question. Can
yes,
you, when you're doing the class and you're like typing the code in,
yeah.
um, oh, I don't know if this one you're going to do in that format, but when you're typing the code in, it's very hard to read the code because at that point it's very, very small text. For me,
Oh, I think I can. I think I can adjust the zoom.
oh, cool, perfect. Yeah, yeah, that was what I was going to ask.
Just remind me when I get over to MATLAB, which I will in a second.
Okay.
Um, but real quick, I want to explain the difference between normal variables and cell variables. And I want to start with a really brief theory moment. I assure you, I'm not going to spend a lot of time. But basically, your computer has RAM, random access memory, and this is where everything that your computer is currently doing is stored. And RAM is a linear array, so it has a bunch of cells in it, a bunch of nodes, and those nodes store binary information. If MATLAB has, a square array, basically it reads the first dimension, then jumps to the second dimension and reads, down again, and then keeps reading down and across. So basically it converts a two-dimensional array or a three-dimensional array or what have you into a linear array. And this is where it gets important to, at some point we're going to talk about the difference between linear indexing and subscript indexing. But for now, just knowing that memory is stored in this linear fashion. One of the things that I want to point out is this is why it can be really important to pre-allocate an array. And rather, like if you're using a loop, you don't want to loop and increase the size of your array as you loop. So for example, if we create an array that's 5, 7, 6, 8, and then we iterate around and then we want to add in another value right here, this memory block's probably already taken, which means MATLAB has to delete this and rewrite it somewhere else in a free region of the RAM space. And then the next loop around, it gets bigger again. It has to delete that and rewrite it in a new region of the RAM space. On the other hand, if you know that by the end of all of your looping, you're going to have an array that's 50 by 50. If you pre-allocate that space, even if you don't know what numbers are going to go in there, when MATLAB first writes that variable, it's going to make sure it has enough space for that entire variable array. The other thing that I want to emphasize is basically you call this variable, and it's a 2 by 2, array, and maybe you call it A, maybe you call it B, maybe you call it Bob, maybe you call it Im. Whatever it is, it's a variable. That name, and for those of you who took CS10, you might recognize this, but that's basically a pointer. And what I mean by that is that variable name is pointing to a start point in this RAM, and then MATLAB reads from there. So the reason why I want to introduce this concept of a pointer is basically what a cell array is an array of pointers. So if you create a cell array, inside the cells, you can have very, very different kinds of information because MATLAB's not actually storing the information in the cell array. It's just storing a list of pointers. So a cell array is basically a variable of variables. You can have a bunch of variables in the cell array, which allows you to have kind of dynamic naming. So let me jump over to MATLAB and let me see if I can increase the zoom. Um, hmm. Oh, wait, there's view. Nope, that's not useful at all. That might be too big. Is that better, Lisa?
Yes, 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 yes. Much better. Thank you.
I'm going to have to remember to turn this back when I'm when I go down to code next time.
It might be good for your eyes to keep it like that.
Oh no, you wanna always stress yourself, always push yourself. All right, so basically clear all, and then so I want to give you an example of where a cell array might be useful, because we're going to get into a situation where a cell array is useful. So I want to kind of prime it first. Let's say I have a variable, and I'm just going to call it Bob1, and Bob1 is 5. But then I'm running a different code, and I have 2 Bobs. So I have, Bob1 equals 4 in this case, and then I have Bob2 equals 7 in this case. So now you can see in my workspace, I have Bob1, Bob2. And if I wanted to, I can use write a script and take advantage of Bob1 and Bob2. But then let's say I need to, a different script where I have 3 Bobs. Well, then I just write out Bob1, Bob2, Bob3. But let's say I'm working on a script where I don't know how many Bobs I'm going to have. One time I run the script, maybe I have 2 Bobs. Another time I run the script, I might have 4 Bobs. So I need a way of labeling the Bobs such that it can change from run to run of a script. So as a more concrete example, let's say I have an image file that has multiple images in it. So normally what we've been working with in the last couple of weeks is 3 TIFFs, red, green, blue. And each TIFF is exactly 1 image. The green image is, a single image plane. The red image is a single image plane. What if instead of, that 1 image, some of these had 2 images, some of them had 4 images? We would want a format where we could open that file and we could get all of the images. But rather than, opening, an image file that has 4 images contained in it as a container, where we have M1, M2, M3, M4, what if we had a single variable image or a single variable I or something like that? But inside that image, we had multiple containers for those different images. And that's where cell arrays come in. What a cell array is, it's a variable of variables. So I can use, rather than Bob1, Bob2, Bob3, I'm going to clear all. And then instead of Bob1, Bob2, I'm going to make a cell array. And I'm specifying going into a cell using curly braces. So inside the cell Bob1 is an array 5, 5, 3, 2. So Bob is now a 1-by-1 cell array that has a 2-by-2 matrix inside of it. And then I could do Bob2 and make that, a single number. So if you think about this, I could store 1 image in Bob1. I could store a second image in Bob2, and these don't even have to be the same size. They don't even have to be the same type. I can have Bob3 be metadata. So I can store different kinds of data inside a cell array, because all the cell array really is, it's a list of pointers. It's an array of arrays. It's a variable of variables. So each one of these individual variables, I could do the exact same thing with Bob1, Bob2, Bob3, but this way I have a single variable of variables that stores all my subvariables in the different cell arrays. So hopefully that makes sense. So the reason why this comes in handy is if we're writing a script and we don't know, or it's not convenient for us to have a situation where we know going in to it how many images we have or how many Bobs we have. We want to provide a generic variable that can open any number of Bobs, any number of images. And then if we want to know how many images we have, we just look at the size of that variable. So the core of that is, and I'm going to save this as demo 2. Demo 2. Okay, so basically, the core of this, what I really want to talk about is the Bioformats toolbox, which is a toolbox produced by the Open Microscopy Environment, which is a project of LOC at University of Wisconsin. And basically, it's an open source toolbox where you can open many, many different image formats, including our format of choice, the Leica image file or LIF. If you're working on the confocal, you have produced LIF files. And MATLAB doesn't open LIF files with ImRead. You need to use an adapter, and that adapter is the Bioformats toolbox. So what happens is if you have the Bioformats toolbox, you can download it from online, and then you can extract it into the directory you're working with. And the key things are this bioformats package.jar file, and then all these bfsave, bfopen, bninit logging, bfgetreader, bfgetplane. All of these things are just M files, functions that you can use in order to work with these kind of complex images. So I want to start with a relatively small image file. So I'm going to create a variable A, and I'm going to use bfopen to open this file, which is, as you can all see, a LIF file. And keeping with the theme of tonight, this is going to be an epithelial stromal co-culture stained with glee. I haven't looked at this image in a really long time, so I don't even know what's in there. But I'm going to go ahead and run this. And what we have stored in A is a 6x4 cell array. And the bfopen command is always going to produce a n×4 array. The first column is going to be every image in the system. The first column is where all the image data is stored. The second column is metadata. The third column is also metadata. And the last column is also metadata. So basically, depending on whether you want the raw metadata, don't really worry about the third column. It's almost never useful. Or the OEM metadata, you can get that from either the second column for the raw metadata or the OME metadata, which is basically just a standardized format. So depending on specifically what you want to do, you can learn about the image. We're not going to get into that today. I don't want to do too much. But the cool thing about this metadata is it stores everything about the collection. So in each one of these metadata chunks, you have the objective that you used. You have the laser power. You have the gain. You have the date and time the image collection started. You have the date and time the image collection ended. You have the user who was logged on at the time. You have all of this information stored in the metadata. You have the XY position of the stage. So were you all the way to the right? or are you all the way to the left? You have all these parameters stored in the metadata. You will be surprised the level of detail that microscopy metadata is stored in. They will store literally everything. And this is good. This allows you, so for example, Maggie was asked the question, did you use the same collection conditions? And she did, and that's the right practice, but let's say you weren't sure. That data is stored in the metadata, that information's in the metadata, and MATLAB can harvest that for you. If you want to know what your pixel size is, it's in the metadata. If you want to know what your z-step size is, that's in the metadata. So all this information is there. We're not going to get into harvesting it. It's a little weird at first, but I just want to say that all of that information is there. So we have this 6x4 array, and that's because this image has 6 series. So a series is a grouping of images. So we have images of images. So things that might represent a series are if you take a z-stack. That will be multiple images together. Let's say you took a z-stack and you had 10 z positions. That would be 10 images. Those would all be packaged up into a series, and that's going to be stored in this first cell. Another possibility is, let's say you have one Z position, but you took 3 channels. So you use 3 different lasers and you collected 3 channels. Then you're going to have those 3 different channels stored together as 3 separate images, but they'll be part of the same series. And if you combine all of these together, so let's say you had 3 channels and 10 Z positions, you would have 30 images all stored as a series. So let's take a look what's inside one of these series. This is actually a very simple file. In order to go inside of a cell, I need to use curly braces, and I just want to demonstrate the difference. I'm going to use A with regular parentheses, A11, because I want to go into the first row and the first column. And it informs me that A11 is a 3x2 cell. It's a 1x1 cell array that contains a 3x2 cell. You can see that A itself is a 6x4 cell array. So I didn't go inside the cell, I just took that cell. And that's an important distinction, and I think that's one of the things that tends to trip people up. I want to actually go inside the cell and see what's in there. As it turns out, it's another cell. But I'm going to use curly braces. So A11 with curly braces. Now I'm going to go inside this 1x1 cell array and then open up what's inside, which is a 3x2 cell. Lisa, you have a question?
Yeah, when you say you took the cell, do you mean you put it in a new, like you remade A into a new, like if you were to name something B and then B is equal to A, comma, 1, 1, or parentheses, 1, 1. Did you just completely like remove what was in that cell and put it in a new location and it's no longer there, or it's still there?
It's still there. I copied it. So for example, if I do B equals A 1, 1 using parentheses, B is now a 1x1 cell array, but A still has that information.
But when nothing was lost because you used a parentheses instead of a curly brace?
No, nothing was lost. I simply took the entire, if you think of the cell as a box, so like a cell array has a bunch of boxes. And then inside each box, there might be something inside the box. Maybe it's an image, maybe it's a number. Using parentheses like I did right here, B equals A, parentheses 1, 1, you're taking the box. You're copying the box. If I use curly braces instead, I'm opening the box and taking what's ever inside the box and copying that. Does that make sense?
So it's like, at that point, it's directly everything that's there instead of the, like, It's like opening the present and putting it there as opposed to like keeping the present unopened.
Yes, exactly.
Okay.
So
okay, I follow.
you're working with the cell array and you have straight parentheses. You're taking that entire present and copying it. If you use curly braces, you're opening the present, seeing what's inside, and copying that. So in the parentheses way, B is a cell array, 1 by 1 cell array. In the curly brace way, B is still a cell array, but that's because there was a cell array inside the cell array. And the reason why I want to explain this is because this kind of nesting can get confusing. But okay, let's go back to A11. This is curly braces. So inside is a 3 by 2 cell array. So I said the outer cell array is going to have that first column is going to be the image data, and then everything else is metadata. Each cell inside that first column is going to be a series of images in the first column, and then in the second column, more metadata. So inside that first column, you're always going to have 2 columns, always 2. The first one is the images, and the second one is the metadata. So just to zoom in on what this metadata might look like, it's not all of the metadata associated with the file anymore. Oops. So I'm going to nest my cell indexing. So A11, and I'm going inside 11, and I'm going to go to 1, 2. So first row, second column. So I now have 2 pairs of curly braces. One gets me inside the first cell. The second set gets me inside the second level of the cell array. And what it tells me is the file name. This is series 1, and this is plane 1 of 3, and it's channel 1 of 3. And if I go to the second row, plane 2 of 3, channel 2 of 3, and hopefully everybody can guess what the third is going to be, plane 3. But okay, so I know what I'm looking at. Let's actually look at it. So let's do, I'm going to create an image, or I'm going to create a variable. The image already exists, it's stored in A, but I want to harvest that image for my own purposes. And I'm going to go to row 1, column 1, and then image sc in. And let's see if this pops up. And this is the image. It's not super bright, I apologize. But hopefully you guys can sort of see that there's something there. We can edit the, wow. My computer does not want to cooperate. We can brighten it a little bit by adjusting the color map. Someday. Maybe not. Oh, there we go. So the color map editor popped up, and I'm just going to bring down the ceiling. So maybe 1000. And, you know, we've brightened the image a little bit. You can kind of see that there's cells there in the background. So this was the first channel. I could also do this rather than assigning it to a, yeah, Nick.
Is there an association with the channels? Like it says channel 2 of 3. Do you know what like channel that is on the confocal, or is there's an association there with like what you're using, or is it just it's 1 of 3?
So in this bit of snippet of information, you know that it's 1 of 3. If you go to the core metadata, so A14, for example, that stores the laser, gain, type, everything for every channel associated with that series. So it'll tell you what your 3 channels are.
Okay, cool. Thank you.
Lisa?
Does this need to be in a specific path? Like where your bfopen, where is it grabbing this lif file from, and does it need to be in a specific place to be able to do all of this?
The easiest way to do this, there's always many ways. The easiest way, and what I strongly encourage, because this minimizes screwing up, bfopen and all of your bfopen M files, your bioformats toolbox, and your lif file should all be in the directory you're using. So that should be in like your working directory, and your script file should be in there. So I do want to point out again, just to go back to Lisa's question, the data hasn't been deleted from A. And we don't need to create a variable in order to do this. I could also just use this in order to display the same image. And this is indeed the same image that we had. If I wanted to instead display the second image, I go to the second row. And this is the second row. And then this was channel 2. And I do a thanks, Zoom. I don't actually remember what all these channels are. We could dig into the metadata and find out. But just by guessing, I would say that this is probably DRAC 7. It's a nice nuclear label. Also, it's the third channel, which tends to be the deep red. We could verify this either by going to the metadata to see what the collection conditions were, or going to, this was taken by Christina. We could go to our lab notebook and see what imaging she had. So this is basically how you open bioformat or more complex data formats, and how you get the images out. The nice thing about this is A is always going to be a n by 4 cell array, where n is the number of series, and 4 is always going to be 4. The first one is image, and then the next 3 are metadata. And then inside that first column of image data, you're going to have a m by 2 column, where m is the number of planes. So maybe it's multiple channels, maybe it's multiple Z positions. All of your image data is in that first column, and then that second column is specific metadata, like which channel this was, which Z stack this was, things of that nature. So that's basically what I wanted to cover. Does anybody have, Lisa? Did you raise your hand? No? Oh. Nick? Anybody? Nope. All right. Any other questions?
I have several questions that are maybe related to this, but it has to do with my specific script.
Sure. I mean, so if nobody else has any questions, people are welcome to stick around while I try to answer Shane's questions, or they can take off. And I will upload this later tonight.
I'm going to get myself a drink of water, and then we'll start if that's okay.
Yep, that's all right. All right, have a good one, everybody.
Thanks, Professor.
Right back.
Yeah.
You there?
Yep.
Okay, so I'm going to go ahead and share my screen here, and then I'm going to open up MATLAB. Okay, so we've talked. So one of the things that I was meant to do